This video is about temozolomide. So, what does it treat? Temozolomide is a chemotherapy drug which is used to treat certain types of brain tumours called glioblastoma multiforme, the most common and malignant glial tumours. The other type of brain tumour is anaplastic astrocytoma, but this one isn't as rapid developing. Initial treatment includes surgical removal, which is followed up by post-operative radiotherapy. So what are gliomas? Gliomas are made of heterogeneous groups of tissues with abnormal growth found in the central nervous system. As I said before, they are malignant, meaning cells and tumours can invade nearby, move from the original site, and spread to other parts of the body to form other tumours elsewhere. So what are some of the causes of developing glial tumours? Genetic factors. The genes your parents give you may mean you are higher risk of developing a glial tumour. Race. Some ethnicities are more susceptible to developing glial tumours over others. The last one is pretty controversial, but it is believed that sulfur use can increase the risk of developing a glial tumour. Moving on to the structure of temozolomide. It contains 6 carbons, 6 hydrogens, 6 nitrogens, and 2 oxygens. It is a relatively small molecule with a molecular weight of 194.055 grams per mole. It has a log P of -1.1. Its small molecule size means it is readily absorbed into the digestive tract. Furthermore, its lipophilic properties means it is able to pass through the blood-brain barrier. Temozolomide contains 1 hydrogen bond donor group and 5 hydrogen bond acceptor groups, meaning it is orally available. It usually comes in capsule form. As you can see from this diagram, the molecule contains an imidazole ring and is therefore in a class of alkylating agents called imidazotetrazine. Temozolomide is a prodrug and is stable in acidic pH environments such as the stomach. However, when it comes into contact with the basic pH of blood and tissue in the body, the molecule is transformed into its active metabolite as it is hydrolyzed and turned into its reactive form. So how was temozolomide discovered? In the late 1970s, in Birmingham, Professor Malcolm Stevens set up a drug discovery laboratory in the pharmacy department at Aston University, where he led researchers to build on earlier work by Cancer Research UK scientist Professor Tom Connors to develop new drug prototypes and test their potential against cancer. Initially, they produced the anti-cancer agent azolastone, but the molecule proved highly toxic to humans. After about a decade of work, in 1987, a potential lead molecule was found, and it was called Monica CCRG81045, and it was this that was then altered over time into temozolomide. He easily convinced the committee to set up the first temozolomide clinical trial on cancer patients in 1980, as there was a lack of potential drug leads since barely any in vitro testing was carried out beforehand to support a lead drug. Now moving on to the development of temozolomide. It all started when Malcolm Stevens became interested in the reactive heterocyclic ring systems, which are rich in nitrogen atoms. This led to temozolomide's nucleus, the imidazole tetrazine's ring system. 2 strands of nitrogen chemistry led to temozolomide's discovery. In 1970, it was recognized that some molecules with a linear chain of 3 nitrogens, called triazines, seemed to have anti-tumor activity. A triazine called DTIC was already on the market against malignant melanoma. It is the prodrug forming the alkylating agent MTIC. The second strand of nitrogen-rich molecules inspiring Stevens were the tetrazine heterocycles. When he arrived at Aston University, the group was exploring bicyclic ring systems, which are fused ring systems which share more than two atoms, and in this case the atom was nitrogen. They explored many fusion of rings, which made the first imidazole tetrazinine, which still contained three consecutive nitrogens, but this time in a cyclic form. After making a series of variations, they formed one containing a chloroethyl group, which showed antitumor activity, which they called a zolostone. However, it was highly toxic to bone marrow, as it cross-linked DNA, and so they went with the only other variant found earlier to have antitumor activity. The other imidazole tetrazine was the methyl derivative, and this was temozolamide, which basically replaced the chloroethyl group with a methyl, and although it was less active, it was not toxic. We will now move on from development onto synthesis of the drug. So, there are two methods of synthesizing temozolamide, and they will be referred to as Scheme 1 and Scheme 2. So, Scheme 1. In this process, molecule A, called 5-amino-1H imidazole 4-carboxamide, is converted into molecule B, which is 5-diazo-1H imidazole 4-carboxamide, which is then cyclized with methyl isocyanate in dichloromethane. This provides a high yield of temozolamide. However, this process requires isolation of the unstable and potentially dangerous 5-diazo-1H imidazole 4-carboxamide, that's molecule B. Moreover, methyl isocyanate is a difficult reagent to handle and ship, so basically the first reaction is not highly recommended. Now, let's move on to Scheme 2. So, the method was adapted firstly in 1994. The agent used was discovered within the same year. Compared to the first synthesis method, the reaction time is pretty long, so this is a disadvantage, and basically it takes up to several nights to complete the process. Yet, there are advantages of using this scheme as well, one of which is that the reagents are a lot more safer compared to those in Scheme 1. Now it's time to talk about temozolamide's mechanism of action. It is a small lipophilic molecule that is an orally available non-functional DNA alkylating agent. TMZ acts as a prodrug, stable at acidic pH, so permits oral administration but degrades above pH 7. The hydrolysis reaction of TMZ produces an electrophile called methyl diazonium cation. The electrophile produced, called methyl diazonium cation, is attracted to the carbonyl on guanine, carbon 6, which is a nucleophilic site. TMZ alkylates guanine at carbon 6 to give O6-methylguanine. O6-methylguanine mispairs with thymine during DNA replication, therefore alerting DNA mismatch repair, MMR. DNA MMR recognizes mispaired thymine on daughter strand and exercises it, but O6-methylguanine persists in template strand. Futile cycles of thymine reinsertion and exertion result in DNA strands breaking, causing replication forks to collapse. Cell cycle arrest is triggered, which occurs in the second cell cycle following treatment, and apoptosis occurs. Methylguanine methyltransferase is a protein that recognizes the O6-methyl group on guanine and removes it, therefore preventing mispairing with thymine to occur. Good response to TMZ therefore requires functional MMR and low levels of MGMT. Most important DNA repair systems impacting the mechanism of action and cytotoxicity of TMZ are MGMT, MMR, and BER. Finally, moving on to side effects. Temozolomide contains side effects similar to many cytotoxic drugs. Cytotoxic drugs affect rapid cell growth. Though it is targeted towards cancer cells, other cells in the body also divide rapidly and are affected by the drug. This therefore causes on-target side effects, which means that the adverse pharmacodynamic effects are linked to the drug's mechanism of action. which, as previously mentioned, is its alkylation of DNA on normal tissues. Off-target side effects are adverse effects that involve different sites of action. The most common on-target side effects are alopecia, hair loss, sore, dry mouth, constipation, diarrhea, nausea, and bone marrow suppression, which leads to an increased risk of infection as the white blood cell count is lowered during the course of treatment due to the cytotoxic drug also affecting their production. The patient's white blood cell levels need to be heavily monitored during the course of treatment for adjustment of doses based on severity of bone marrow suppression, and the other common side effects are fatigue and headaches. Tumor lysis syndrome is an interesting side effect. It occurs due to treatment-related rapid destruction of tumor cells. This then leads for the tumor to then release their contents into the bloodstream. This causes metabolic disturbances in the body and leads to other side effects such as hyperuricemia, which means an increased level of uric acid in the blood. That is also an off-target side effect. So what have we learnt? Temozolamide is a chemotherapy drug which treats brain tumours called gliomas. It was found due to Malcolm Stevens' interest in nitrogen atoms. Its lead molecule was Monica CCRG81045. MTIC is the active form of the drug, and temozolamide is the methyl derivative of azolastone, which makes it not toxic. In synthesizing temozolamide, safer reagent methods are preferred. It works by mispairing with thiamine and causing apoptosis. Finally, it is a cytotoxic drug and has many of the same side effects.